Welcome back, everyone, to today's Explainer. Today, we're unpacking a truly fascinating discipline that has completely revolutionized how we understand life itself. I'm talking about bioinformatics. Seriously, if you've ever wondered how modern biology manages the absolute avalanche of genetic and molecular data generated every single day, well, this is the ultimate solution. Today, we're going to demystify how digital code is helping us decode biological life. Let's get into it.
Okay, let's dive into this. Here is our roadmap for today. We'll be defining bioinformatics, looking at the evolution of biological data, exploring the core components, navigating the databases, checking out the tools of the trade, and finally, decoding sequence alignment.
Section 1. Defining bioinformatics. This is the critical intersection where computer science meets biology. So, bioinformatics isn't just one single thing. It's really a highly integrated branch of science. We're talking about bringing together computer science, math, statistics, chemistry, and engineering. Why? Well, for the storage, retrieval, and analysis of biological data. It's basically what happens when we use computational software to actually make sense of raw biological data. We're taking the sheer, messy complexity of biology and analyzing it through the rigorous, structured lens of mathematics and computer science.
Now, the goal here isn't just hoarding information. Sure, developing databases for efficient storage, access, and management is absolutely crucial, but building the algorithms and applying analytical tools is where the magic actually happens. Scientists are constantly developing new algorithms and statistics to assess these complex relationships among massive sets of data. Then they apply specific tools to really interpret it all. It's a super active, dynamic process of discovery.
Moving right along to Section 2, Evolution of Biological Data. Let's see how a slow manual process urgently demanded a digital revolution. Let's move to the timeline and see how this builds. Back in 1965, Margaret O. Dayhoff established the very first protein sequence database. Pretty groundbreaking stuff. But look at what happens around 1977. DNA sequences start accumulating rapidly in the literature. Why? Because it became way easier to predict protein sequences by translating sequenced genes rather than sequencing the proteins directly. Boom! The sheer volume of uncharacterized proteins just exploded. This massive data dump forced scientists to create digital libraries. We're talking GenBank at NCBI and the EMBL in 1980, the Protein Information Resource in 84, and the DNA Databank of Japan in 86. Managing all this rapidly accumulating sequence data urgently required new software, which fully birthed bioinformatics as a specialized field by the mid-1990s.
That brings us to Section 3, the core components. Let's shift our focus and break the discipline down into its three fundamental pillars.
Listen, you simply cannot have modern biology without these three sequential steps working together in perfect harmony. First, you have the data. That's the raw biological information itself. Second, the database. These are the computerized storehouses. And third, the database mining tools, the actual software used to extract meaning. If you're missing even one of these pieces, the whole system just breaks down.
So let's zoom in on that first component, the data. What exactly are we storing here? There is an incredible variety of raw data getting fed into these systems every single day. We're talking nucleic acid sequences, your raw DNA, cDNA, RNA, ESTs, but it goes way beyond just genetic code. We've also got protein sequences and their complex 3D structures, metabolic pathways, gel pictures, and even the scientific literature itself.
Now, what's really interesting about this slide is just how staggering this number is. 30 million. That represents the amount of scientific abstracts housed just within the PubMed bibliographic database alone. That's just one database, providing links to full articles, related text, and GenBank entries. It really shows you how deeply interconnected and vast the literature component of biological data truly is.
With all that data, we definitely need a place to put it. Section 4. Navigating the databases. The digital libraries of life.
Databases are the absolute heart of bioinformatics. A database is a vast, computerized storehouse of records in an electronic environment. But it's not just a digital dumping ground, right? It relies entirely on standardized formats. That standardization is the magic key that allows scientists to effortlessly extract, add, and merge specific records from this chaotic sea of raw nucleotide and protein sequences.
When we talk about nucleotide sequences, there are three major global players. You have GenBank at NCBI in the USA, the EMBL in Europe, and the DNA Databank of Japan. What is incredibly cool here is the global cooperation. These three massive databases collaborate under the International Nucleotide Sequence Database Collaboration. They're updated and exchange data daily. That means accession numbers stay consistent, and researchers worldwide are working with the exact same unified genetic information without any legal restrictions.
Now, for proteins, the databases get highly specialized. You have primary databases like SwissProt, which don't just store sequences, but actually provide high-level annotation, describing the functions, domains, and post-translational modifications. Then there are secondary databases like ProCite and Prints. These look for specific sequence patterns and fingerprints to see if a new sequence belongs to a known family. And finally, you have primary archives like PDB and SCOP, which serve as the actual structural blueprints for the physical 3D structures of these macromolecules.
Section 5. Tools of the trade. So the data is vast. The databases are perfectly organized. How do scientists actually search through them? Let's check out the mining tools.
And this brilliantly illustrates how specific tools act like highly specialized search engines, totally tailored for genetic code. Without these, those massive databases would be pretty much useless. You've got BLAST from NCBI, which is used to analyze sequence info and detect homologous or related sequences. There's Entrées, also from NCBI, which lets you seamlessly access literature, sequence, and structure databases all in one go. And tools like DNA Plot out of the UK, functioning specifically as a sequence alignment tool.
Section 6. Decoding sequence alignment. This is arguably the most fundamental operation in all of bioinformatics.
So what is sequence alignment? Basically, it's arranging DNA or protein sequences to identify regions of similarity. By literally lining up the code, researchers can see if there's a functional, structural, or evolutionary relationship between different organisms. Think of it like taking two sentences, putting them side by side, and seeing if they were copied from the exact same original source document.
There are two main types of alignment to wrap your head around here. First is pairwise alignment. This is lining up exactly two sequences to assess maximum identity and homology. It's your foundational starting point. Then you have multiple alignment, which is exactly what it sounds like, lining up 3 or more sequences simultaneously for a much broader comparison across multiple organisms.
So the crucial point is how we actually measure if two sequences are related. We look for similarity. But in bioinformatics, similarity isn't just some visual guess. It's a very specific mathematical combination. In this field, similarity actually equals identity plus conservation. You literally have to add both together to truly understand the relationship between the biological data.
Let's break that down just to make sure the nuance is totally clear. Identity means the sequences are exactly invariant. The code is identical. It hasn't changed at all. Conservation, on the other hand, means there has been a change at a specific position over evolutionary time, but, and this is key, that change still preserves the physiochemical properties of the original residue. The code changed, sure, but it still performs the exact same function in the resulting protein. Together, identity and conservation give us the real measure of biological similarity.
Which leaves us with this final thought. We've seen bioinformatics evolve from a few uncharacterized sequences to a daily updated global network processing an insane amount of data through complex algorithms. With millions of new DNA and protein sequences being generated every single day, what hidden evolutionary secrets will bioinformatics unlock next? It's a dynamic digital toolset that is quite literally writing the future of biology. Thanks for joining me on this explainer, and I'll catch you in the next one.